Let's take a quick look at DNA replication through a survey of the associated proteins. DNA replication starts in a very specific place in the DNA identified by a certain sequence of bases. It's often called the origin of replication. The first protein, let's call it promoter 1, spins up and down the DNA looking for the right DNA sequence. When it finds the right sequence, it stops, binds on the DNA, and brings in, let's call it promoter 2. Promoter 1 and promoter 2 together form a pre-replication complex. The pre-replication complex brings in our third protein, which is helicase. Helicase melts the DNA and separates the two strands. The melted DNA does not want to be in two strands. Its lowest energy form is double-stranded. And our fourth protein is designed to deal with this. It's called single-stranded binding protein. As the DNA is unwound, it wants to form kinks in the DNA. The DNA is a spiral, and you can't unwind it without there being strains on the molecule. Our fifth protein is designed to deal with this, and it's called gyrase. The gyrase wraps around the DNA at the replication fork, breaks one of the phosphodiester rails of the DNA, and allows it to spin against the other one, allowing the DNA to spin around, relieving this strain. DNA is always replicated 5' on the trinucleotide to 3' on the existing DNA. So DNA cannot be replicated from nothing. There has to be a primer. The primer is added with primase. So once it gets to this point, the regular DNA polymerase can come along and start doing its job. Its job, of course, being to add trinucleotides, 5' on the trinucleotide to the existing, 5' on the trinucleotide to the existing one, and goes about its business. This is the main DNA polymerase. The primase puts on a primer that is made out of RNA. It's not made out of DNA. It works as a primer, but it's not made out of DNA. So there's a different DNA polymerase that goes to the primers, chews them off, and again 5' to 3', trades them out for DNA. So there's two different DNA polymerases. When the DNA polymerase is done, it's hooked them together, 5' to 3', 5' to 3', but there's a nick in it that the DNA polymerase can't fix, and that's taken care of by ligase. So this is now our 9th gene product. And then finally, the process of DNA replication cannot copy the DNA all the way out to the end of the chromosome. So the very ends of the DNA kind of are left out. Our last enzyme is telomerase. And the telomerase adds, it's a riboprotein combination, and it adds nucleotides to the ends of the DNA. And this is a summary of the proteins involved with DNA replication.